Hello, it's Dr. Rafael Gutierrez. Today I'm going to make a video that I will use for people who want to watch PowerPoints when I lecture in class. Now this does take a different approach than the other presentations I've been doing with the models. The reason is this is actually set up for a full semester course. And so it will be a little different than what you would actually see in, let's say, a general anatomy class. So the first thing I actually do want to tell you about is the body actually has something called bilateral symmetry. Bi means two, lateral means side symmetry, meaning if we were to take a straight line, you could see one here, down the middle, which is called a mid-sagittal line, and you cut the body straight down that mid-sagittal line, you would end up having two relatively equal sides, mirror images of each other, which means you have bilateral symmetry. Now, when you look at that, the sagittal plane divides things from the right side from the left side. And as I mentioned that, I should also mention something called anatomical position. If you notice, the palms are facing out, the palms are facing out, toes are pointing forward as well, face is forward, and this is anatomical position. I want you to write this down if you're taking the class. Any question asked in anatomy assumes that the body is in anatomical position. It doesn't matter your position, it's the body that you're looking at, and it's going to be an anatomical position. So know your anatomical position. So over here you see a sagittal plane. It separates the right from the left, and the other thing that it does is it gives you a center point, a midline. And anything that's closer to the midline is usually referred to as medial. Anything away from the midline, going to the sides here, would be lateral. So if you actually take the sternum, it's medial compared to the shoulder, which the shoulder would be lateral compared to the sternum. And these things will change depending on what you're looking at. Now, besides the sagittal plane, you also can take the body and draw a line right over the skull, the head, right where you would have if you were wearing a crown or tiara, you would see the main portion. So in Spanish and Latin, that's called corona. So the plane that actually follows from right to left across the body, separating the front from the back, would be the coronal plane. And coronal plane separates things that are in front, which would be anterior, things that are behind, which would be posterior. And so we start getting some direction of the body. So we have medial, lateral, anterior, and posterior. And we also have something called a transverse plane, which is a belt-like cut across the body. These words are actually going to come across over and over again, so I recommend writing those down and knowing them. The transverse plane pretty much separates the top part from the bottom. And so you have superior and inferior. Now with the arms and legs, you also have things away from the area, which would be distal, and that's different from lateral, which if you think of lateral being where the thumb is in anatomical position, the pinky would be medial, and you would actually have distal being if you're looking at the palm, it would be the base of the palm would be proximal compared to the farther end of the fingers, which would be distal. Legs actually work the same way. And so it's just one way to actually look at the body. Now, if you notice, there are some roots that are important to know. Bi meaning two, lateral meaning sides or away from the midline, sim meaning same, metri meaning measurement, dorsal technically means top, ventral means bottom. You have medial, which would be towards the midline. And I actually do want you to know thoracic because we're going to talk about that a lot coming up. And you also do have proximal and distal, which we'll talk more about later. Now, we do have a couple divisions of the body. If you look here, you see the cranial cavity, where the brain goes, and the spinal cavity, where the spinal cord goes. You also see the thoracic cavity, the abdominal cavity. Now, the way we develop, and we'll deal with this later, the back actually develops on top of the head, on top of the organism, and the front develops on the bottom. If you look at cats or dogs, you'll notice that they have that structure. So the cavity that makes up the cranial cavity and the spinal cavity together is called your dorsal cavity. Dorsal meaning technically top, and it does work because if you look at where the dorsal cavity starts, it is the top. Ventral cavity is in front, and it has a couple different divisions. The thorax is the chest area, and inside you have a cavity called the thoracic cavity. Underneath it, you have a muscle called the diaphragm that separates the thoracic cavity from the abdominal pelvic cavity. And there's a line, the true pelvis here, which separates the abdominal cavity from the pelvic cavity. And so you have different body cavities that we are going to deal with in anatomy. It is important to know these cavities, and it's actually kind of neat in that if you get into both of these cavities, you actually have space between the cavity and you have a membrane, and the organs in there kind of float around, which we'll talk about more later. Okay, so we've talked about planes and directions. Superior meaning above, inferior meaning below, anterior in front, posterior behind, medial meaning to the side, medial meaning to the middle, lateral being to the sides. Superficial would be, it's actually outermost. It doesn't matter if it's outermost posterior or anterior. If it's closer to the surface, it's superficial. Deep would be obviously deep. Proximal, I talked about, which is closer to the midline, the trunk. Distal would be farther away from the trunk. Dorsal being technically being top, but a lot of times it's used as posterior. Ventral meaning bottom, but a lot of times it is used in anatomy as anterior. I mentioned bilateral symmetry, which you know means has two equal sides. And I mentioned sagittal, which is actually, if you think of Sagittarius, it's the symbol has a bow and arrow. Sagittal is a path an arrow would fly, so you have mid-sagittal, which separates the right and left. You have parasagittal, which are any cuts along that. Coronal, meaning crown, which would be the cut which separates the body in anterior and posteriors. And the transverse, again, is a belt-shaped cut. Now over here I do have a general picture of a cell. I found it online. It supposedly is fair use. And you can see a lot of the different things in a cell. Now, every cell will be a little different, but it will have the same organelles. And the reason is because every cell is going to need certain things. For instance, if we look here, we can see the nucleolus, which is where we end up finding the genetic material of the cell. A lot of people think genetic material is just for cell division. That's part of it. But it's also the blueprints for every protein that's available in the body, whether it's intracellular, inside the cell, or extracellular, outside the cell. So we have the codes for the protein inside the nucleolus, and we have the nucleus around it, which is a phospholipid bilayer with transmembrane proteins, which allow mRNA molecules to move out of this area. They do allow certain things to come in as well. And they're called nuclear pores, the transmembrane proteins. If you listen to the name, transmembrane, trans means across, membrane is a phospholipid bilayer of the nucleus. And so if you have the DNA, you copy it into mRNA, and it will leave the nucleus through a nuclear pore. Now, you'll see a lot of proteins inside here, which are these little dots, which are called ribosomes. What ribosomes do is they take the mRNA, which we got from the copying the DNA into mRNA, and it translates it into protein. What it does is it takes the three codes of the RNA, and it binds them to a specific RNA molecule called a transfer RNA, which it carries one type of amino acid. And so the ribosomes take those amino acids and glue them together to form a protein structure. Now, the protein structures made by the free ribosomes here will usually stay in the cell. And sometimes they'll actually form these filaments in here, which make something called a cytoskeleton. Cyto means cell. Skeleton is skeleton. And so a lot of the cytoskeleton proteins in here are made from the free ribosomes. You also do have some protein structures within there, which are enzymes. Usually they'll have a suffix ASE, which is ACE, which tells you it's an enzyme. It's a protein catalyst, and it's going to do something. One of the main ones that you'll actually find is ATPases. ATP is a chemical, adenosine triphosphate. ACE is an enzyme that works on that. And so what happens is you take ATPase with the, you take ATP, it binds to the ATPase, and the ATPase can sometimes break it. And when it breaks it, it releases energy. Now, not all the proteins you want are going to be in the cytoplasm here. Cyto means cell, plasma, fluid. And so we do have another structure. You can see the rough endoplasmic reticulum. You also have a smooth endoplasmic reticulum, which is a little different. The rough endoplasmic reticulum has ribosomes attached to it. And what happens is when you want to make a protein that's going to be bound to a phospholipid bilayer or is going to be in a little bag, a vacuole, so it doesn't actually break anything apart, you're going to make it in this rough endoplasmic reticulum. Same thing, the mRNA goes to a ribosome. The ribosome will go to the endoplasmic reticulum, bind to it to start making the protein. And then from the rough endoplasmic reticulum, it will go to the Golgi, and the Golgi will be able to modify it a little bit, and then it'll be a vacuole waiting here to be excreted. It pretty much going from this cell out. And that is going to, what's going to happen is the phospholipid bilayer, which makes a little vacuole, is going to fuse to the membrane of the cell membrane, which is a phospholipid bilayer as well. And it's going to leave the cell as a process called exocytosis. Exo meaning outside, cyto meaning cell, osis a process. So let's say you're making a protein that you wanted to use to form a structure around the cell. So it would go from the nucleolus, the DNA would actually be copied into RNA, the RNA would leave, go to the ribosome, go to the rough endoplasmic reticulum. From the rough endoplasmic reticulum, it'd go to the Golgi would then do some modifications, put in a little vacuole, and the vacuole would leave the cell through exocytosis, and so you'd make a protein that goes outside the cell. It doesn't matter what the protein is, if it's a digestive protein, if it's a protein that is going to be laid out to give structure around the cell, or if it's a protein that's going to be a messenger protein to send out to other things. It's going to use the same process. So we've talked about the nucleus, the rough endoplasmic reticulum, the Golgi, and we also do have a smooth endoplasmic reticulum. The smooth endoplasmic reticulum does not have the ribosomes. It is actually a lot of times modified to either make fats and steroids, or store fats and steroids, or store calcium. You sometimes have calcium ATPase, which is a protein enzyme which takes calcium and actively pumps it into the smooth endoplasmic reticulum. That will be important a bit later. Now there are certain things in here which are lysosomes, which lyso means to break, somes are just a container inside. Peroxisomes, which are vacuoles which actually have peroxide in there, which are used to destroy different things that aren't supposed to be in the cell. You also do have a lot of other cytoskeleton proteins. For instance, the centrioles are cytoskeleton proteins which help on cell division, which we'll talk about later. You also have mitochondria. Mitochondria, everyone talks about it as being the powerhouse of the cell. But to truly understand it, you have to understand how the cell gets energy. Simplest way to look at it is with sugar. Sugar comes into the cell and it's broken into a molecule called pyruvate. Now, whenever you break something, it releases energy. When you break glucose into pyruvate, you release energy, and you have an ATP, a enzyme that takes the ADP, adenosine diphosphate, and a phosphate, and it puts it together to form a adenosine triphosphate, ATP. And that's how your cells are going to store energy. So if you need energy for something in the cell, you're going to take that ATP, break it into ADP and phosphate, release the energy, and use it. Now that gives you about 2 ATP per glucose, which isn't a lot. But you can take the pyruvate, modify it, and send it to the mitochondria, which the mitochondria has a lot of great things. One of the things it does have is it has its own DNA, so it could replicate on its own. And what it does is it takes the products of breaking glucose, the pyruvate, takes it in, and through a lot of different processes, it will usually take a lot of oxygen, break down the pyruvate into carbon dioxide and water, releasing a lot more energy and using that energy to make a lot more ATP. So usually when sugar goes all the way through with the mitochondria to carbon dioxide and water, you end up getting about anywhere between 32 to 48 ATPs rather than just the 2 that you ended up getting by just breaking the sugar here. So to run the mitochondria, you do need oxygen, you do need to start with sugar, and any other energy source will actually go into the mitochondria to be broken down. Fats, for instance, can actually be broken down in mitochondria using oxygen. And so we have the basics of the cell. You do have other structures. You have cilia, flagella, microvilli, a lot of different things that can actually come into the cell, but that's the general principle of the cell. So there are roots that you should know. Cyto, or site, meaning bag technically, but in this case we're dealing with the bag, the cell as being a bag. Endo being inside, exo being outside, trans, again, going all the way through. So when we deal with transmembrane proteins, they're proteins that go all the way through the cell membrane. And so now that we have the basics of the cell, I'm just going to talk a little bit about the cell division, mitosis. Usually your cells are not going through mitosis unless there's damage to them, if they have to replace it. There are some cells that do go through mitosis more often than not because of their exposure to the environment. But usually your cells are not going to be growing once you are developed. But when you're developing from a giant cell into a organism, you do go through this rapid cell division. And one of them is called mitosis. And mitosis, you have cell division, but you maintain the number of chromosomes. You start with the interphase, you go to prophase, you go to metaphase, anaphase, telophase, and then you have 2 cells. And the faster this happens, the smaller the final daughter cells become. If you're constantly going through a growth phase, you can actually maintain a big cell. When you fertilize the oocyte, the cells actually get bigger and bigger because of that. When you are developing, you have a giant cell and it goes through rapid cell divisions, and it doesn't necessarily go through a growth phase because you need the cells to get smaller because of osmosis and diffusion. But interphase is where your cells mainly are going to be. And then one of the things a friend of mine used to say is, if you want to know the phases, remember I, P, and then on a mat, M-A-T. So you have interphase, I, prophase, P, and then mat, metaphase, A, anaphase, telophase, and then you end up with 2 daughter cells. And the idea is you have the same number of chromosomes at the end. Now there are different things. There's prophase 1, there's metaphase 1 and metaphase 2. I'm sorry, there's mitosis 1 and mitosis 2. And what it does is it just makes bigger, smaller and smaller cells. And you do also have meiosis, which is for cells that are eventually going to be sex cells. But big thing I want to deal with is how we develop. And I got this picture online. It was all the pictures I've got are online. They're supposed to be fair use. If one of them is incorrect, please let me know, and I'll redo this without making it. So in embryology, you make a morula. Morula technically means mulberry, which is pretty much a berry. And so you start with the 1 cell, and you notice it divides into 2. It would then divide into 4 and continues going. And so you look at this, it looks kind of like a blackberry. The cells get smaller and smaller as you go through this cleavage. Eventually you make something called a blastocyst. Again, image is supposed to be fair use. But what you see is the cells around called the tropoblasts and the cells inside called the inner cell mass. And you have a fluid-filled cavity called the blastocele. And the cells that are actually going to become the organism are just the inner cell mass. All these other cells are going to actually become temporary structures. So by week 2, our pretty much cell mass has actually come up here and made 2 cell layers. And you have the 2 layers actually have cavities. One has an amnion up here, and the other one has a yolk sac here. And these two cell layers are in a ball, so it's called a bilaminar disc. This actually happens at the end of the week 2, and up until this point, the cells don't really have too much development. When you start having the epiblast and hypoblast, epiblast would be the one up here, hypoblast the one down here, you start having some specializations of cells, and that will come into play later. So week 3, by the end, cells from the epiblast come between the epiblast and hypoblast, and so you end up making 3 cell layers, trilaminar disc. You have the ectoderm, the mesoderm, and the endoderm. And this continues, where you end up pulling in part of this. You end up forming something called a notochord inside the mesoderm, and you start forming a tube here, which is going to be the neural tube. So you have the outer layer, which is going to be called the ectoderm, the mesoderm, the middle layer, and then you can see the tube here, which is going to be your endoderm. And at this point, you start forming organs. Up until this point, the cells are really not specialized, and even these are not completely specialized, but they're starting to get specialized enough that they may not be able to make every type of tissue. So there's a lot of different things these cells will form. Each germ layer, each cell layer will form a different tissue. Always remember the roots. Bi means 2, like a bilaminar disc, 2-layer disc. Derm means skin. So you have the epiderm, outer layer, outer skin, mesoderm, middle skin, and endoderm, which is the inner skin. It's a tube. Hypodermis you also have, which we'll talk about a little bit. And now I actually just want to tell you about how the body is organized. And so what you have is this. You have the cells. The cells make certain things around it, which is a matrix. It's usually anything that's in a tissue that's not a cell. So you have cells come together to form tissues. And you have 4 basic types of tissue, which are your connective tissue. Usually, these will have more cells than matrix. I say usually because adipose tissue is a connective tissue, but it tends to have a lot of cells, and the matrix is actually within it. You have the epithelium, endothelium, and mesothelium, which is usually a bunch of cells with a little bit of proteins holding it together, which would be their matrix. And these usually line organs, the outside of the skin, the inside of the mouth, the inside of the lungs, the inside of blood vessels. So these are the lining areas. And then you have contractile tissue. Contractile tissues, just like it says, it contracts. And there's only 3 contractile tissues, no matter what anyone tells you. There's striated skeletal muscle, striated cardiac muscle, and smooth muscle. And the last one is neuro, which is the nervous tissue. And we are going to see a lot of these together. Now, one thing that happens a lot of times is these different connective tissues will come together to form organs. So the organs are a bunch of tissues working together for a particular function. And you have different organs. For instance, the heart is an organ, and you have different tissues in the heart. Lining the inside of the heart, you have a endothelial layer. You also have connective tissue between it, but you also have a connective tissue around the heart, dense irregular connective tissue. You also have contractile tissue, which is the striated cardiac muscle, and you have nervous tissue going with it, which are the neurons that go and tell the heart to either speed up or slow down. So you can see how organs can have multiple different tissues. Now, organs come together to form organ systems, and these are just systems that work together for a common function. A lot of times, though, these organ systems interact with each other because certain things have to happen. So, for instance, the cardiovascular system does work with the nervous system. It does work with the digestive system. It works with a lot of different systems, and so these systems, while we can't separate them out a bit, you'll notice that there are interactions between them. And those interactions are because of the following, of what we're going to talk about, which is the organism, which is pretty much you. And so that should be the end of lecture 1. I will talk more about tissues so you can see what I want you to see on each tissue. I hope you enjoy this and have a nice day.